Good afternoon. My name is Janelle Cedillo, and today I'm excited to introduce the keynote speaker, Dr. K. Tai. Dr. K. M. Tai is a professor and Wiley Vail Chair of the Systems Neuroscience Laboratory at the Salk Institute, and an adjunct faculty member at the University of California, San Diego. Her research program focuses on understanding the neurobiological mechanisms underlying social and emotional processes at the circuit, cellular, and synaptic levels, particularly those relevant to psychiatric disease. Professor Tai was born in Ithaca, New York, and graduated from Ithaca High School in 1999. She began her professional training at the Massachusetts Institute of Technology and graduated with a major in brain and cognitive sciences in 2003. After taking a year off to travel, she earned her PhD from the University of San Francisco in 2008 and trained as a postdoctoral fellow at Stanford University from 2009 to 2011. She then became an assistant professor at MIT in 2012 and was promoted to associate professor with tenure in 2018. She then moved her laboratory to Salk Institute in 2019. Professor Tai has been recognized with a number of prestigious research awards, including the NIH Director's New Innovator Award, the Presidential Early Career Award for Scientists and Engineers, among others. She has also been recognized with a number of awards for mentoring at the undergraduate, graduate, and postdoctoral level. Further, she is committed to outreach, promoting diversity and inclusion in science. When I was in high school, I had the pleasure of working with Dr. Tai as a summer intern in her lab. I firmly believe that she is one of the most influential women in STEM today. I also highly encourage those who are interested in pursuing a career in STEM to begin internships in high school so that you can meet amazing, highly motivated, and just overall great people to be around, like Dr. Tai. A great program I recommend checking out is the Hightoff Brody Summer Scholars Program. More information on the Salk's website for that. But without further ado, please welcome Dr. Tai.
Hello, can you guys hear me? You guys see my slides? I'm okay?
Looks great, thank you.
Thanks for responding. Okay. Hi, everyone. Oh, what a wonderful introduction from Janelle. I miss her. And it's so great to be here to talk to you guys. So today is a little bit of a different talk than I normally do, so please bear with me if there's some hiccups. But today I'm going to tell you just really briefly about who am I, the person behind the science, and how I got to be here. And something that I'm really interested in right now, which is the science of social isolation and what is happening to our brains when we're socially isolated. And that's the main parts of the talk today. If I have time at the end, I'd love to say a few things, more casual comments about emotional associations, bias awareness, and some general advice for young investigators like yourselves. So, okay. You guys can laugh if you want to, but this is me in high school with my little sister. And in high school, so I was really blessed and extremely fortunate to have amazing role models for parents. Both of my scientists, and my mom was a biologist, a scientist. And there really were very few women scientists. And so I had amazing role models, and I was so sheltered. I didn't really realize how privileged I was until I went into the world. But in high school, I didn't really think of anything about science. I never really thought I was going to be a scientist. I understood my parents did something that they were really into, but I didn't really understand what they did. So I played sports. I was really into sports, and that was probably like the main thing I was into in high school, and like going to parties and stuff like that. In academics, so like I'm sort of breaking up, my personal life and my professional life. And in high school, I was really into English literature and art and poetry, and applied to like most colleges as an English major. When I went to college, I got really into running and doing outdoor stuff. I ran a marathon. I got really interested in urban dance, breakdancing, or bowling is what we call it. And then this is the first time I got really interested in neuroscience research. It was really early. Mostly I was just taking classes in neuroscience. So I wasn't that sure that this was going to be, my calling in life. I had a few research opportunities, but they weren't. It wasn't obvious to me that this was what I was supposed to do. So I did some, also still into poetry and art, and had some. Oh, this is a picture from the top of this rock climbing wall. But yeah, it was really balanced. I didn't really necessarily know exactly what I wanted to do. I thought maybe I wanted to go to grad school, but I wasn't really sure, and I wanted to see what else was out there. So I took a gap year, and I had one kind of not great experience with, it was sort of an isolated incident, and then I wasn't sure. I thought maybe I'm not good at science, and maybe I don't have what it takes. And so I tried a bunch of other things, and I just traveled and backpacked for my gap year, and I was a yoga instructor, and I tried to write a novel. That's what I thought I was supposed to be doing in my life, was like, English literature novel type thing. That was really hard. Actually, it was very lonely, difficult. I was a dance instructor. I tried to play professional poker. I just explored other career paths and realized actually, work is hard sometimes. And, that I tried a lot of different types of ways to make a living. And, I eventually came back to science. So had a lot of fun, and it was great to have those experiences. But I think what I didn't get from being a dance or yoga instructor that I wanted to get from science is that I wanted to feel like every single thing I did mattered. And, I could just teach the same yoga class like over and over and over, and nobody seems to notice. Nobody says anything. So, it kind of just felt like it doesn't matter what I do each day. And, of course, I really enjoy yoga. Yoga is still a big part of my life. But I wanted something where I would create something that would last, that other people could build on, that would change how other people think and how other people do science in the future. And I really like the feeling that science is always moving forward, that we're building upon, the work that you've already done or other people have done, and you're just part of something bigger. And I really appreciate that. Oh, this is my pet lamb that I had in my gap year. Her name's Biscuit. Okay, so then after my gap year, I went to graduate school at UCSF, and then I did my postdoc at Stanford. And during this time when I was living in the Bay Area, I got really, really into breakdancing and b-girling. I was a competitive breakdancer for about 10 years. That was a huge part of my life. And I was also really like snowboarding. And, this is me watch battle. This is me with my crew. We used to do halftime shows for the Warriors, the Golden State Warriors. They weren't as good as they've been recently, but yeah. And then, of course, I was also really into neuroscience. And the first year of graduate school, I had a tough time. I wasn't sure. I almost dropped out of grad school. I was just like, oh my gosh, everybody knows more than me. Everybody's smart, and I don't fit in here. But then, I just had a really amazing PhD mentor, Patricia Janik, who just believed in me. And, that her looking at me and just genuinely believing in me that I could do it made me think I could do it too. And I don't know what I would have done without that type of mentorship and support. So I was really grateful for that. And I'm always still involved in outreach. That's just something I enjoy. And oh yeah, this is just more pictures. This is my crew. We just battled these girls in. And we had an all-girl B-girl crew, which is very unusual. Like, breakdancing is extremely male-dominated, more so than science. And so, I guess I feel very comfortable. And I wanted to say that, a lot of people think it's weird that you would be into breakdancing and also science. Like, those things are really different, but it's not that different. In both cases, you've got your crew, your lab, your team that you work with. You practice together, you prepare, you create something, and then every now and then there will be a conference, a scientific conference or a jam, a battle, and you go and show everybody what you've been working on. And, you've got to protect your reputation and rep for your crew. So that part's similar. And then after that, I went to MIT as an assistant professor. I started my family. I have 2 babies. They are now no longer babies. They're 5 and 7. And I still have a lot of hobbies. I love surfing now that I've moved to San Diego. My kids have gotten me really into skateboarding and taekwondo and other things. And I think it's just really important to remember that you can be more than one thing. And I can be a scientist. And so, okay, that's the who am I part, which feels kind of funny. But and then the next part is something that I think is really important. So needless to say, this is July 4th, 2019. 2020. New Year's Eve, 2019, 2020. And social isolation and perceived loneliness correlates with a number of deficits in mental and physical health, including shorter lifespan, in terms of mental health disorders, increased anxiety and depression, also increased morbidity and mortality for both cancer and heart disease. increased inflammation, and increased symptom severity with a viral immune challenge. So all of these, negative health consequences, it's really important to understand the neural circuit changes that underlie, the experience of social isolation or perceived loneliness to prevent the negative effects. And so for universally, across social species, social isolation is considered aversive. And so a few years ago, a brilliant postdoc in my lab, Jillian Matthews, did an experiment. And I know some of this is a lot of technical stuff, so you just stay with me. But she did an experiment that was supposed to be about something else, and her control group looked different. And it was supposed to be a cocaine study. Her control group looked different. The animals were injected with either cocaine or saline, which is just salt water, and then put in a cage alone. And then we wanted to see what plasticity happened in their brain. But her saline group, her control group, also changed, and that was weird. And so after more experiments, and I really applaud Jillian, because I think a lot of scientists would have gotten that result and just been like, oh, I don't know, my experiment's not working, something weird's happening, and like give up. But instead, Jillian trusted her data. She followed her data and figured out what was causing the plasticity. And it turned out it wasn't the cocaine. It was actually the social isolation. And so these dorsal raphe nucleus dopamine neurons are different than a lot of the other dopamine neurons people usually talk about. Usually people talk about dopamine neurons and they mean VTA dopamines, ventral tegmental area dopamines. Like if they're, 99% of papers about dopamine and motivated behaviors is about these other type of neurons, but there's also these dorsal raphe nucleus dopamine neurons. And when you isolate an adult mouse for 24 hours, they will undergo synaptic plasticity onto these dopamine neurons. These neurons will become more active, which we can read out with a genetically encodable calcium indicator, which just means it glows whenever there's more calcium in the cell, and the cell has more calcium whenever it's active. So whenever it glows, the cell is active, and we can see what activates the neurons. And basically, we found that these dorsal raphe nucleus dopamine neurons are more active in response to a social target after social isolation than they are when they're group housed or to a novel object. And so this suggested that dorsal raphe nucleus dopamine neurons are sensitive to acute social isolation and can track a change in social. So we next wanted to use a technology called optogenetics, which just allows us to control different neurons in whatever way that we want. We can make them active and we can excite them, or we can inhibit them and make them silent. And so we wanted to see first, and this is a genetically encodable light-sensitive protein. That means when we shine light into it, we make the neurons fire. And so this is a technology that was invented about 16 years ago and has transformed neuroscience. But basically, when we activate these dorsal raphe nucleus dopamine neurons, we saw that we produce an increase in motivation, an increased social contact. However, when we silence these neurons, or when we activate these neurons, they were more social and they would seek more social contact, but it was also aversive. And so this was very confusing because usually when you think of dopamine, like dopamine signals, it would increase sociability, and also it's positive. Like you think of, for example, cocaine, which is a drug that targets the dopaminergic system, and it increases sociability, but it's positively reinforcing. And animals and people may work hard to get this positive reinforcer in the case of substance use disorders. But here, these dorsal raphe nucleus dopamines are totally different. They made animals not like the photostimulation. They would not lever press or do any sort of thing to get photostimulation. They would try to get away from it instead. And so this was kind of confusing initially. How can something be pro-social and unpleasant? But if we compare this concept to our feeding work, we know that, you can eat a delicious dessert at the end of dinner. You don't really need it. You're not hungry, but it's just so delicious that you enjoy the food reward. Alternatively, you can seek food for a negative valence reason. Like maybe I'm just so hungry. I feel like, I'm just I'm like, my body's like shutting down. I'm so hungry. And I'll dig to the bottom of my backpack for some, nasty fiber bar or whatever, because I want to escape the unpleasant state of hunger. And so those are two reasons you can eat: for the reward or to escape the unpleasant state of the need. Similarly, you can engage in social interaction because of the social reward. It just feels great to socialize. Or you can engage in social interaction and seek social contact to escape the unpleasant state of being lonely. When we silenced these neurons, when the animals were living in a group, nothing happened. But many animals, humans, rodents, monkeys, if you isolate them for a short period of time, like 24 hours, and you reintroduce them to the social group, there will be a rebound of social interaction. Oh, let's stay up all night and catch up, that, more affiliative behavior, etc. When we isolate animals and then silence these dorsal raphe nucleus dopamine neurons, we block the isolation-induced rebound of social interaction. And so this together is sort of looking like maybe these neurons are the cellular substrate of loneliness. And this was really exciting because no one had ever discovered the neurons that, mediated the experience of loneliness before. But there was a lot of criticism because this is not a word that we really encounter in neuroscience. It's more of like a psychology term. And so, if you think about the definition of loneliness in psychology, it's something that increases the motivation to seek social contact. Got that? It is aversive and unpleasant. It's a need state. Got that? It's distinct from generalized anxiety. I didn't show you those experiments or data, but we also did those experiments, and it is distinct. And then the fourth one's really tricky. The fourth criterion of being lonely is that it's subjective. And that just means you can feel lonely at a party or like in a crowded street, but you can also feel not lonely alone in your home. And so all these things really matter. It is subjective. It's a very different experience to be kicked out of a party than to decide to leave on your own, right? And so this subjectivity issue is really hard to get at in mice, and even in people, but especially in mice. So how do we get at this? And so what we found was that all mice form a social hierarchy in their cages. And there'll be an alpha, the dominant mouse. There'll be a subordinate mouse too. And we just thought about it like, maybe this is a way to get at subjectivity, because the experience of a dominant mouse is going to be very different than the experience of a subordinate mouse. A dominant mouse, enjoys its social environment. It has prime access to resources. It can mate with all the females. Life's good for the dominant mouse. And so when it's isolated, maybe it feels more lonely, and a subordinate feels relieved when they're isolated because now they have all their bullies are gone and they're not being deprived of any prime resources. So we tried to think about this, and we wanted to look also to see if this was true in humans. And so in collaboration with Livia Tomova and Rebecca Sachs, we looked in humans and found a similar effect. So obviously it's not cellular resolution in humans, but it is a similar effect that you get a craving signal in social isolation when you're presented with social stimuli as you do when you're presented with food stimuli after food deprivation. Okay, so I'm going to skip this one part just to get to this one concept. So there's one thing that's really interesting. And the big question I have right now is, why is it that after acute isolation, animals of all kinds will when reintroduced to the social group, there will be a rebound of social interaction, pro-social. It's a very positive valence stimulus to be reintroduced to your social group. And so with chronic isolation, it's the opposite though. After you reintroduce animals after chronic isolation to the social group, they are the opposite. They are territorial, they are aggressive, they are avoidant, and engage in and basically display antisocial behaviors. So what gives? How is it possible that the same manipulation, social isolation, and the same stimulus, reintroduction to the social group, can either good or bad in these different conditions? And so we've come up with a conceptual framework. And on here, the y-axis, we have the quality and quantity of social contact. Here is, this is time. Let's say here is when an individual was socially isolated. their effector system, this part of the circuit that helps just drive increased motivation to seek social contact, is activated. And then that serves to close this deficit, and then the whole social homeostat can shut off once you've returned to your normal baseline. But what about when you can't? For example, say you're socially isolated. Now your effector system is activated, meaning you feel lonely. You have an increased motivation to seek social contact. You are engaging. But after a certain amount of time, or maybe a certain amount of failed efforts, there is a set point adaptation, a change in the set point of the desired amount of social contact. And then once you go into a chronic isolation state, then the set point kind of adjusts to what you've got, this new normal, and so to speak. And so once you've adjusted your homeostatic set point, if you were to be reintroduced to your previous optimum, you might perceive that as a surplus. And so this can provide one possible explanation for how it is that the same social isolation and then reintroduction to the social group can flip valences from being positive to negative at different time points. And so now the key is to understand the neural mechanisms that underlie this particular transition. So I'll end there. I want to thank all the wonderful people in my lab that are currently in my lab, as well as a bunch of very important alumni and everyone who's ever worked with, my collaborators, my funding sources, and you guys for listening. And yeah, I can stop and take some questions if we have time.
Thank you so much, Dr. Tai, for giving us a talk about your research, something that's so poignant that a lot of students and people have experienced firsthand with the pandemic. We do have a couple of questions for you. So the first one was submitted in advance, and it is, as a principal investigator, do you still get to do experiments? And then if you could kind of just expand on that a little bit, tell us what a day in the life looks like for a PI.
Yeah, that's such a great question. And so I think the thing about being a principal investigator, the best thing about being a scientist and being a principal investigator is you get to do whatever you want. I don't have a boss. I can run a lab that's small where I do experiments, or you can run a lab of any size and do experiments. Some PIs like doing experiments and they choose to do experiments. Some principal investigators will prefer to do less of that. So it depends. I think I'm sort of, right this moment, I'm not doing experiments, but I've done experiments when we first moved to Salk, and it was fun to get back into the lab. But what I mostly do is I meet with people in my lab. I help them design experiments. I supervise the performance of the experiments. We look at the data together and interpret the experiments together. And then I communicate the science and, of course, think of new ideas and apply for new money to do new research in the future. But a huge part of my job is sharing with the world what we found in papers that we write up, in talks like this one. So that's kind of what I do most of the time. It's kind of a choose your own. You can make your scientific research program whatever you want it to be, and you can even change it. Like I used to have a bigger lab, and then now I have a more medium-sized lab, and so I like it better that way. So it's just up to you.
Awesome. So we have another question that kind of touches on earlier in your talk. You mentioned how important your PhD mentor was for you, but this question is, what characteristics do you look for in a mentee?
Oh, that's a great question. I think it's different. I have all sorts of different types of mentees with all sorts of different backgrounds and like levels of experience. I think the things I would look for in a mentee are motivated, ethical. Actually, I should have said that first. Integrity and ethics are like number one. If you are, if you're not ethical, then science isn't the right career path for you. But ethical conduct. And then motivated, communicative, open, curious. Those would be like things that I would like to see in a mentee. But yeah.
Awesome. And kind of along similar lines, what advice would you have for a student that is considering a career in science but isn't sure if it's right for them?
I mean, I was exactly like, that in that place. And I honestly wasn't really sure if I was going to stay in science until like midway through grad school. And then something clicked for me. And like, it was like the first time I designed an experiment all by myself. Nobody told me anything. I just thought it up in my head and I did it and it worked. And I was like, oh, I could just do this. Like, oh my gosh, because I'd done a lot of experiments that other people designed and it didn't work. And I was like, I don't know if I'm going to be able to do this. Like, my stuff doesn't work. But, and lots of stuff doesn't work. But the feeling that it worked just gave me a lot of hope. And that moment it all clicked for me. But I would say that, you don't know if you're going to like something or if it's right for you until you try it. And I think, what does it take to be a scientist? I think the most important thing is being really curious about how things work. And that's kind of the most important thing, just being curious about things. And then all different types of people, all different types of personalities, there's so many diverse scientists that are very successful. There are many ways to be successful. So I think you just have to try it and feel it out. But I liked it. It's something where you can be yourself and you decide what you want to spend your time doing.
Yeah.
I like that.
I totally agree. I think it's really important to hear diverse voices in science to really be able to go further and push boundaries, and we need that. So for students that aren't sure, don't get discouraged, continue to stay curious. Thank you so much, Dr. Tai, for providing us a wonderful keynote that's so relevant for everyone. We're so grateful for you. Thank you again, students, for joining us. And if you're joining us for a laboratory later this afternoon, we'll see you later.
Thanks, everyone.
Bye. Thanks, everybody.